Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Jake Scott. He's a clinical assistant professor of medicine and infectious diseases at Stanford. So we're going to talk about his work there. So, Jake, thanks for coming.
Yeah, thanks for having me. Appreciate it.
Yeah. So tell me about your work in infectious disease, and I'm sure it includes COVID, but tell me about your work up until the beginning of this year, and then now what's happening.
Sure. Yeah. So I primarily work as a clinician, as a doctor seeing patients. Most of what I do is see patients who are hospitalized with various infectious diseases or complications that could be infectious diseases. I also see patients in my office in the outpatient setting with a variety of issues. So in the outpatient setting, one of my areas of focus has been HIV. So I help with the diagnosis and management of patients with HIV. I also do HIV prevention, so using pre-exposure prophylaxis. And in the hospital, I see just a really wide variety of infections. And so when I tell people that I work as an infectious disease doctor, even if I tell, close friends, I think most people assume that probably just pertains to sort of exotic contagious diseases like Ebola or, COVID-19. But a lot of what I deal with are very common infections. So urinary tract infections, pneumonia, bad cases of appendicitis, diabetic foot infections. So I basically help I serve as a consultant and help primary doctors in the hospital. So the hospital doctors, they call themselves hospitalists, or the ICU doctors or the ER doctors. So they'll call me and ask me to help diagnose the patient and get them on the right antibiotics if they warrant antibiotics.
Beyond a regular doctor diagnosing someone and giving them antibiotics, it seems like it's still, for the most part, I guess, is just broad spectrum, nuclear bomb type antibiotics. I mean, are we able to do rapid sequencing and figure out a particular pathogen and then do a targeted antibiotic, or what's the protocols that you're doing lately?
Yeah, so it's always going to be a case-by-case basis based on my clinical impression. So it's not always broad-spectrum antibiotics. And oftentimes, a big part of my job is talking doctors out of using antibiotics. The overuse of antibiotics has been leading to a disastrous situation where multi-drug resistant organisms have been steadily on the rise. So a huge part of my job is what we call antimicrobial stewardship. And that just refers to protecting the precious antibiotics that we have. But again, yeah, it totally varies depending on the patient. So sometimes the ER will call me in the middle of the night if I'm on call and say, yeah, I've got a patient who has metastatic cancer, and they've got this and that and all sorts of, various complicating conditions. They're profoundly neutropenic, so their white blood cell count's really low, and they're going to the ICU in septic shock. Well, in that case, I'm definitely not going to take any chances, and I'm going to use broad spectrum antibiotics. But another very important part of my job is tailoring antibiotics as quickly as possible. So to answer your question about rapid diagnostics, yeah, there's definitely been a lot of progress in the last, I'd say 10 years or so, where we're able to more promptly identify the particular pathogen. So, for instance, The ER, if they see someone who's in septic shock, they always get blood cultures. So then the blood is sent to the lab, and the tech in the lab will put the blood on some cultures, which would basically just provide nutrition for any bacteria that are in the blood to help grow the bacteria. And once there's growth, then we can rapidly identify the particular organism using pretty advanced technology. So we can get an identification of a bacteria, within hours of someone arriving in the emergency department. So if they say, the blood cultures grew E. coli, and I've got someone on medication that cover, the whole spectrum of bacteria and maybe fungi as well. Well, then I can, even if they're very sick, I know it's E. coli, so I can take, 2 of the antibiotics off and just keep them on one. So, yeah, but I can't emphasize enough how important it is for me to get patients either off of antibiotics if I don't think they need it, and to get them on the right antibiotics as soon as possible.
Well, which antibiotics do you have to be more careful prescribing, and for what reasons? Are certain ones really fast? Do they trigger really fast adaptation to being resistant to them and then to being resistant to other ones? Like, how do you know what to give someone first and not give?
Yeah, great question. So it all depends on my clinical impression as to where the infection is coming from. So if I'm convinced that it's a skin infection because their leg is red and swollen and there's pus, then I would know that the most likely culprit would be a gram-positive bacteria like Staphylococcus aureus or Streptococcus. And I would then know that I probably don't need to cover for E. coli and a number of gram-negative bacteria. So I would use the narrowest antibiotic I could. There are differences in terms of the barriers to resistance among classes of antibiotics, there are ways in which we prioritize antibiotics in terms of really trying to avoid resistance to certain classes. So, for instance, there's a class of antibiotics called the carbapenems, and they've been well known for decades now for being the broadest spectrum antibiotic. So they cover gram-positive bacteria like staph, they cover gram-negative bacteria like E. coli, and they cover anaerobic bacteria like Bacteroides fragilis. But a huge problem in the last 10 years or so has been this worldwide increase in the number of bacteria that are resistant to this class of antibiotics. So the carbapenem-resistant bacteria are extremely problematic and a huge threat. So for that reason, the carbapenems need to be used very judiciously and should be, I think, in my opinion, restricted to approval by an infectious disease doctor. So when it comes to, I think you touched on a really important sort of misconception about infectious diseases. So, and as an example, I distinctly remember when I was an internal medicine resident in the ICU, while rounding, which is where all the doctors in training circle around the attending or the teacher, and the attending physician will ask us questions and put us on the spot. And I distinctly remember the ICU attending asking all of us what we were going to go into in terms of our subspecialty, and I said infectious diseases. And he said, "Oh, so you're just going to be okay with recommending antibiotics for the rest of your life?" And obviously, I think he realized he was being overly simplistic, but there is this misconception that infectious disease doctors just recommend, the broadest spectrum antibiotic. But coming up with broad spectrum antibiotics is very easy. Any ER doctor, for instance, could tell you, a combination of antibiotics that would cover most of our bases. So like vancomycin and zosyn is a very common cocktail. So you wouldn't need an infectious disease doctor to recommend, the the most, big gun, broad spectrum antibiotics. Our job is, I think, much more complicated than that.
I don't know. Is phage therapy used or recommended very much? If so, why not? What's the state of the art here? What's next and why? How can we make improvements into current protocols?
Good question. As far as phage therapy, I think that there's potential. It certainly hasn't been used clinically yet. And I know that you've interviewed some other folks who specialize in drug resistance, and I don't want to repeat what they've said, but it is worth just sort of reiterating that the growing rise of multi-drug resistant organisms is an enormous threat and extremely complicated. But one of the main reasons why this is an uncontrolled threat is because our market system for supporting research and development for these agents is completely inadequate and has failed us. So when it comes to developing new antibiotics, it's been sort of a lost cause, or at least it's been seen by drug companies as a lost cause, because these are antibiotics that we want to use as little as possible, right? So to tell a drug company or some research organization, hey, help us come up with the newest and greatest treatment for these drug-resistant infections. Well, then they'd say, well, okay, so I'm going to invest $1 billion into R&D for this agent, only to then say, hey, use this as little as possible.
Right,
there's just, yeah. So and I think this is becoming common knowledge increasingly, but so I think that it doesn't necessarily have to be as sophisticated as coming up with an entirely new sort of approach to treating infectious disease organisms. I think that we need to just even work on simple antimicrobial development. There was an antibiotic developed that was FDA approved last year called plasmomycin, and it was a relatively small company, Achaogen, that developed this, and a lot of us were really pleased with it. It seemed like a very promising drug, but earlier this year, or maybe late 2019, the company went bankrupt because so here's an innovative antibiotic that could potentially be life-saving, and the company that developed it couldn't survive. So I think why? Because there just wasn't profit in it, because newer antibiotics are even more precious than the ones we're losing. We don't want to lose the newest and greatest, and I'm not implying that plasmomycin was necessarily the newest and greatest, but once we come up with a new antibiotic, organisms could quickly develop resistance. So no matter what we come up with at this point, there's always the potential for resistance. So we need to use them as sparingly as possible. And
has anyone bothered to look and see why or how? What are the predominant mechanisms by which bacteria become resistant? And are there certain types of molecules that they get resistance to faster than others? And what if you look at the associated virome or phagome, I'll call it, with a given bacteria? Maybe there you'd find more leverage against it instead of just an antibiotic. Maybe you alter its. I'm going to call it the phagome, because multiple phages seem to prey on a given bacteria. Is anyone even looking at that?
Yeah, I think that there are hundreds, if not thousands, of people researching the mechanisms of antimicrobial resistance. People are dedicating their careers to it. A lot of the funding, unfortunately, is insufficient, I'll say. But I do think that there are a number of different approaches that we need to invest in, whether it's phage therapy or CRISPR. I think that alternatives to the penicillin-derived antibiotics, I think alternatives definitely need to be looked at, and we need to take this extremely seriously. You've probably known that there's a UK-based study, I believe last year, that did some very sort of advanced modeling and predicted to come 2050 that antimicrobial resistance was going to be one of the biggest killers in the world and kill more people than cancer. And so this has been, I mean, COVID-19 is obviously just terrifying and a total nightmare, but it kind of came out of the blue, even though, you know, a lot of specialists who were always saying it's not a matter of if, it's a matter of when. But that's something, you know, these sorts of pandemics will hit us periodically, but antimicrobial resistance has been, we've been aware of this ever since penicillin was discovered. You know, Alexander Fleming quickly realized after penicillin was being used that antimicrobial resistance and superbugs, I mean, they were even using the term superbug back then. We've been well aware of this problem, and I feel like it's been not taken seriously enough, and is going to only get worse. So yeah, we need to look at every possible approach. It's definitely going to take an all hands on deck approach, and we've got to get creative and look into all sorts of, you know, viral phage therapy solutions and all that. I don't have any area of expertise in phage therapy, so I can't really speak to that. But I will say that until we have any sort of breakthrough where we're able to use something other than the traditional antibiotics that we've used since the 1940s, that the best we can do is use the current antibiotics appropriately and really curb their overuse. And sometimes, you know, sometimes we look for or sort of expect these breakthrough solutions. You know, like with COVID-19, I feel like a lot of us are just, you know, riding this out until there's some vaccine developed at record speed, you know, or some novel therapy or even repurposed therapy that will save us. But oftentimes it's much simpler than that. You know, like with COVID-19, I think the best we could do is just all wear masks. With antibiotic resistance, the best we can do is not overuse the ones we have.
Well, in terms of antibiotic resistance, what's an example? How do you use an antibiotic in the right way versus the wrong way?
Yeah, so great question. So if I wanted to promote resistance, I would use an antibiotic at a low dose for a long period of time. Okay. So then, you know, that would just be a total setup for an accelerated sort of natural selection of resistance. So the bacteria that, you know, our bodies are full of billions and trillions of microscopic organisms. And if those organisms are exposed to antibiotics, then while they're replicating, there are going to be mutations that confer resistance, and those genes could then spread to even different microorganisms, and resistance can quickly take off. So the best way to avoid promoting resistance is, well, the best way would be to not use antibiotics. But of course, sometimes we have to. And when we do have to use them, the best solution would be to use the most narrow spectrum antibiotic. That's the best antibiotic for that particular infection. And it usually is one. I mean, sometimes we have what we call polymicrobial infections, where it's a group of bacteria, for instance. But for the most part, we're dealing with one bad apple, you know, and so we've got to come up with the most targeted antibiotic for that. and ditch all the others that the patient might be on. And we need to make sure it's the right dose and the right duration. And the duration is key. So we are learning more and more these days that shorter is better, and that we have often been prescribing excessively long courses of antibiotics.
What about cocktails of them? Why does it just have to be one? Why can't you use, a third of the dose of 3 different ones? Why not do that?
Well, then you could lead to resistance to all 3 of those agents.
But how do you determine that calculus? How do you know when that would be better? Because it would, there's 3 different ways that it's kicking the ass of all the microbes there and, and killing them. And so there's less chance to become resistant because now there's 3 different attacks. Maybe they'd have to do an efflux pump to fight one. They'd have to change their cell wall to fight another. They'd have to, maybe if you hit them up with antibiotic different mechanisms, then it's more likely.
Yeah, no, that's a great question. And sometimes that might work. So, you bring up a great question. So something called synergy, or combination of antibiotics going to be better to treat certain infections, like Pseudomonas. And it's been studied a lot. And for the most part, it hasn't shown to be better. Although I think that in theory, a combination of antibiotics can sometimes protect each antibiotic from resistance. So, for instance, there's an antibiotic called rifampin. That's one antibiotic where we would almost never use it alone, other than in latent tuberculosis. Otherwise, resistance develops much more quickly. So we use rifampin in combination with other antibiotics. But a lot of what we deal with are relatively simple organisms that just require the right antibiotic for the right amount of time. So E. coli, for instance. E. coli makes up about 75% of the bacteria in our colon, and it's the most common cause of urinary tract infections. And I see a ton of E. coli urinary tract infections, whether it's simply isolated to the bladder and causing something called cystitis, which is simple bladder infection, or a kidney infection, something called pyelonephritis, or since the kidney is right there at the interface between the blood and urine, it gets into the blood and causes a bloodstream infection and sometimes septic shock. So, I mean, I probably see dozens of cases of septic shock a year caused by E. coli. And we are able to identify pretty quickly in the lab which antibiotics that E. coli is resistant to, and we're able to make sure that they're on the best and most narrow spectrum antibiotic, and it works. And it usually works very quickly, which is one of the things I find so satisfying about infectious disease. I can cure someone and save their life with one antibiotic.
Okay. So in addition to good stewardship and maybe fast sequencing so we can see which antibiotics are appropriate, any idea on what some of the other breakthroughs might be and when they, anything that's close or we just don't know?
Well, in terms of like new drugs?
Yeah. I mean, is there even, from what I've heard, that there's not even, the incentives are not there, the right incentives are not there to create new antibiotics, which is a problem in itself, it sounds like.
Yeah. No, that's a really good point. I think that we definitely need to be doing a lot more as a society when it comes to promoting awareness around antimicrobial resistance and the importance of the appropriate use of antibiotics. So we need to make sure that it's common knowledge amongst everyone that if you have a runny nose and sore throat, antibiotics are not going to help, and they're only going to promote resistance among the bacteria that live inside you and aren't causing any problems. So we need to raise more awareness about resistance. We need to incentivize companies and research institutions to focus on this, because it's increasingly problematic and increasingly deadly, and the current structure is not working. So, there have been a number of attempts to help incentivize companies with various bills, et cetera. I don't know all the intricacies of that, but I think that there's a lot more work that needs to be done in terms of making sure that the antibiotic pipeline is keeping up with the organisms. And these organisms, they've been ready to be resistant to anything that we throw at them for thousands or millions of years, and that's the thing. They've got the home court advantage, and they outnumber us, a billionfold or more. So this is a lopsided battle, and we need to do more and not less. And have you heard of that study of the Amazonian tribe? I might not pronounce it right, but the Yanomami.
Yeah.
Have you heard of that? The study of their microbiome was deep sequenced. You heard about that?
I've heard that they've been studied, but I don't know what the implications were. Everyone just said,
yeah. So I think it's really interesting. So there is a tribe in the Amazon. I believe it's called the Yanomami or something along those lines. And they had been completely removed from any contact. They'd been completely isolated, and they had not been in contact with the rest of the world. And they had never been given an antibiotic. And so someone had, or a team of researchers were able to get fecal samples and do genetic studies looking at their microbiome. And there were two things that were interesting. One, their microbiome was the most diverse. And that wasn't, that surprising, since they hadn't been exposed to antibiotics. They had the most diverse microbiome. But the other thing that I think was much more interesting was that they actually found a number of genes in the bacteria that were in their stool that had potential to encode resistance to a number of antibiotics, including some of our latest and greatest and synthetic antibiotics. So the potential to lead to resistance, it's there and it's built into these organisms. And when we throw antibiotics at these bacteria that are in our guts or on our skin or anywhere else, then that is just adding fuel to the fire. And I just can't emphasize enough that they've got a major advantage over us, and we need to be doing more.
Well, very good. Jake, what's the best way for people to get in contact and ask you questions and learn more? If people have questions about antibiotic resistance, if they want to read maybe papers that you've put out, if you have, if they want to find out more in general about the subject or about your particular work, where can they go? What should they Google you, a Google Scholar, or what's a recommendation? Yeah,
I mean, I don't want to provide my direct contact information. I've been quite busy, especially with COVID-19 and patient care. But there are a number of great resources. So the CDC is a great resource for antibiotic resistance, as is the World Health Organization. They both have a lot of information online. And I think that there's more and more great reporting out there about this issue. There are a number of other institutional resources. The University of Minnesota has a great resource online. It's called CIDRAP, the Center for Infectious Disease Research and Policy. The Infectious Disease Society of America, IDSA, is also a great resource. So there are a number of great resources out there about antibiotic resistance, and it is something that I'm obviously passionate about and love to talk about. So I'm happy to talk more about it anytime. So,
okay. Well, very good. Well, Jake, thanks for coming on the call. I appreciate it.
Yeah, no, it was my pleasure. Thank you.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.